You put down your fork. You push back from the table. The meal is over. And in that exact moment, without any instruction from you, without any conscious decision on your part, your body initiates one of the most complex and precisely timed biological sequences it knows how to execute. A cascade of hormonal signals, neural responses, enzymatic processes, and cellular communications that unfolds over the next 20 minutes with the precision of a choreographed biological program. Most people think digestion is what happens after you eat, but the 20 minutes immediately following your last bite are not digestion. They are something far more important. They are the window in which your body decides what it actually experienced during that meal and what it is going to do with everything that just arrived. Understanding this window changes how you think about hunger, satiety, cravings, overeating, and body composition in ways that no calorie counter or diet plan ever could. Let's go through it minute by minute. In the first 2 minutes after your last bite, your mouth is still active. Salivary amylase, the digestive enzyme in your saliva, is still breaking down any remaining starch. But something more important is happening simultaneously in your stomach. The stretch receptors embedded in the walls of your stomach, mechanoreceptors that respond to physical distension, are sending real-time signals up your vagus nerve to your brainstem. These signals are not about nutrition. They are purely mechanical. Your stomach does not know what you ate. It only knows how much physical volume is present. This mechanical signal is the first layer of your satiety system, and it is the fastest. But here is what almost nobody understands about this signal. It is not telling your brain that you are full. It is telling your brain that food is present. Fullness, the subjective sensation of being satisfied and not wanting more food, requires a completely different set of signals. Signals that have not even started yet. Between minutes 2 and 5, the real hormonal cascade begins. The cells lining your small intestine, specifically the enteroendocrine cells distributed throughout the intestinal mucosa, are detecting the chemical composition of what is arriving from your stomach. They are identifying proteins, fats, and carbohydrates through specialized receptor proteins on their surface, and they are responding by releasing a suite of hormonal signals into your bloodstream. The first and most important of these is GLP-1, glucagon-like peptide 1. This is the hormone that pharmaceutical companies spend billions of dollars trying to replicate with drugs like semaglutide and tirzepatide, the same compounds behind Ozempic and Wegovy. Your body makes it naturally every single time you eat, provided you eat in a way that triggers its release. GLP-1 does 3 things simultaneously. It stimulates your pancreas to release insulin in proportion to the glucose arriving in your blood. It slows the rate at which your stomach empties its contents into your small intestine, extending the digestion window and reducing the speed of glucose absorption. And it travels through your bloodstream to your hypothalamus, where it begins activating the appetite suppression circuitry that will eventually tell your brain the meal is complete. But GLP-1's effect on your hypothalamus is not instantaneous. It takes time. The signal is traveling through your bloodstream, crossing the blood-brain barrier, and binding to specific receptors in your arcuate nucleus. This process takes approximately 15 to 20 minutes from the moment food first reaches your small intestine. That gap, that 15 to 20 minute lag between eating and satiety signaling, is one of the most consequential biological facts about human nutrition, and it is almost never discussed. Between minutes 5 and 10, a second hormone enters the picture. Cholecystokinin, or CCK, is released from the upper small intestine in direct response to fat and protein content. CCK has a more immediate effect on appetite than GLP-1. It travels faster through the nervous system via the vagus nerve rather than exclusively through the bloodstream. It signals to your brainstem that fat and protein have been detected, that the meal has nutritional substance, and that gastric emptying should slow. CCK is one of the primary reasons that meals containing adequate protein and fat produce more lasting satiety than carbohydrate-only meals. A bowl of plain pasta triggers very little CCK release. A meal containing salmon, olive oil, and vegetables triggers a robust CCK response that actively suppresses appetite through your nervous system within minutes. The difference in satiety between these two meals is not psychological. CCK is also responsible for triggering your gallbladder to release bile and your pancreas to release digestive enzymes, the downstream processes that will actually break down the fats and proteins over the next several hours. But its appetite-suppressing signal is immediate and measurable. By minute 10, if your meal contained adequate protein and fat, CCK is already communicating with your brainstem that substantial nutrition has arrived. Between minutes 10 and 15, your blood glucose begins rising in earnest. The speed and magnitude of this rise is determined almost entirely by what you ate and in what order you ate it. If your meal began with fiber from vegetables, the physical mesh that soluble fiber creates along your intestinal wall is now slowing glucose absorption dramatically. The same amount of carbohydrate that would spike your blood sugar sharply if eaten alone is being released gradually into your bloodstream over a much longer window. Your pancreas is receiving a steadier, smaller glucose signal. It is releasing insulin in proportional pulses rather than a single large surge. This controlled insulin response is the difference between a metabolism that efficiently clears glucose and one that chronically overproduces insulin, drives fat storage, and eventually loses sensitivity to insulin signal altogether. Insulin sensitivity, the responsiveness of your cells to insulin signal, is not a fixed trait. It is rebuilt or destroyed meal by meal through the size and frequency of your insulin responses. Every meal you eat in the right sequence, with fiber first, protein and fat second, carbohydrates last, is a meal that protects your insulin sensitivity. Every meal you eat in the wrong sequence, carbohydrates first, on an empty stomach, with nothing to slow their absorption, is a meal that chips away at it. Between minutes 15 and 20, the final and most powerful layer of your satiety system finally activates. Leptin, the hormone produced by your fat cells in proportion to your total body fat stores, has been circulating in your bloodstream throughout the meal. But leptin's effect on appetite suppression requires the cooperation of GLP-1 signaling in your hypothalamus to become fully active. As GLP-1 levels finally reach the receptors in your arcuate nucleus around minute 15, your hypothalamus begins integrating all of the signals it has been receiving: the mechanical stretch signals from your stomach, the CCK signal from your vagus nerve, the GLP-1 signal from your bloodstream, and the background leptin signal from your fat cells, into a composite assessment of how much you ate, what you ate, and whether enough nutrition has arrived to consider the meal complete. This integrated signal is what you experience subjectively as feeling satisfied. Not just physically full, satisfied. The urge to continue eating is gone. The food in front of you loses its appeal. Your interest in the meal ends naturally. This is your satiety system working exactly as it was designed to work. And it takes a minimum of 15 to 20 minutes after your last bite to fully arrive. Here is why this timeline is the most important nutritional fact most people have never been taught. If you eat faster than this system operates, if you consume enough calories to significantly overshoot your needs in under 15 minutes, your satiety signals will arrive after the damage is already done. You will not feel the overconsumption until you are already uncomfortably full. Research consistently shows that fast eaters consume between 20 and 30% more calories per meal than slow eaters eating the exact same food. Not because fast eaters have less willpower, because the satiety signaling system has a hardwired 15 to 20-minute delay that cannot be bypassed by any amount of discipline. Eating slowly is not a personality trait. It is a biological strategy for staying within your body's actual caloric needs. by allowing your hormonal feedback system to deliver its message before the meal ends rather than after it. This is also why the post-meal walk is so biochemically powerful. Walking for 10 to 15 minutes immediately after eating does not just help with blood sugar management through GLUT4-mediated glucose uptake in your muscles. It extends the eating to satiety timeline into a physical activity window, which means your GLP-1 and CCK signals are still rising while your muscles are pulling glucose directly from your bloodstream. You get the double effect of hormonal satiety arriving fully and mechanical glucose clearance happening simultaneously. Blood sugar stays flatter, insulin stays lower, the satiety signal completes properly, and you are far less likely to reach for something sweet an hour later, because the cravings that drive post-meal snacking are almost always the direct consequence of a blood sugar spike and crash caused by a meal that was eaten too fast for the satiety system to catch up. The 20 minutes after your last bite are not empty time. They are not the passive aftermath of eating. They are the most hormonally active window of your entire digestive cycle. The window in which your body builds its complete picture of what just happened, decides how satisfied you should feel, determines how aggressively your pancreas should respond, and sets the metabolic tone for the next several hours. What you do in those 20 minutes, whether you eat dessert immediately, reach for your phone, lie down on the couch, or take a short walk, shapes that picture. Slow down your eating. Put the fork down between bites. Eat with people in conversation rather than with screens. Build fiber, protein, and fat into every meal before carbohydrates arrive. Walk after you eat. They are targeted interventions in the exact biological mechanisms that determine whether your 20-minute post-meal window leaves you satisfied, stable, and burning efficiently, or hungry again in an hour and storing everything you just consumed. Your body built this system over millions of years. It works perfectly when you work with it. The only thing that breaks it is going faster than it was designed to go. If this changed how you understand hunger and eating, subscribe to this channel. Every video here goes inside the real mechanisms, so you stop guessing and start understanding. Like this video and share it with someone who eats fast and wonders why they are always hungry. The answer has been running on a 20-minute clock the entire time.